Our next speaker is Johan van Vijenberg, a senior scientist at the Lab of Clinical and Epidemiological Virology at the Rege Institute for Medical Research in Leuven, Belgium. He will present on the design and preliminary results of a patient-driven clinical trial in a Belgian long COVID cohort. Welcome, Dr. van Vijenberg.
Thanks for the kind introduction, and thanks also, Danny, for a perfect immunological introduction, which will be very helpful. So good morning, good afternoon, good evening, everyone around the world. So I would like to take you through of this really patient-driven and also, if I may add, data-driven design and some very preliminary results of what we tentatively call a grassroots clinical trial in an ongoing real-world situation, which is a Belgian long COVID cohort. So we've just had a perfect introduction from Danny. This is also a very nice overview recently written by Eric Topol. But I think I can agree with Danny saying that most of these hypotheses actually overlap, and the common thread here is the immune system. So usually when we look at the immune system, we conveniently sample the blood. So, however, we've talked a lot about biomarkers. So what I feel is missing in long COVID, I mean, several people, of course, have proposed this, is first of all, a diagnostic biomarker. And one of the major reasons to look for it is the viral persistence. Of course, you need to biopsy in the right places. There is neuroinflammation going on. Some people have very bravely donated their cerebrospinal fluid. Others have had respiratory samples, got biopsies have been talked about. But I do believe a blood sample is just more convenient for large scale, especially populational studies. We also need, of course, biomarkers to guide our treatments, whether they are antiviral, anti-inflammatory, or anticoagulant, or even new ones that are arising. And then finally, we definitely need the predictive biomarkers. When we look at the patients and their images, we look at their PROMs, so patient-reported outcome measures. So we do want to see the kind of biomarkers before treatment that will help us predict their clinical evolution. And as I said, blood, I think, might be an ideal candidate for these biomarkers because we want them preferably non-invasive, but also we want them to be affordable. Of course, I'm also a fan of 15-color flow cytometry and very rare populations using tetramer technology, but I don't think in a normal clinical setting that's very affordable. So we opted for transcriptomics. And why transcriptomics? Because we do believe it's a very flexible way of measuring how your body, especially your immune system, is responding at that time. We know our DNA, so genomics are fixed. Every cell in our body has the same amount, exactly the same DNA. On the other hand, the RNA that is being transcribed, altogether called the transcriptome, is highly dynamic. And in this case specifically, it can give us a snapshot of the ongoing host and viral interplay while also being tissue specific. Of course, we are also exploring several other people with us worldwide, proteomics and cell-based assays, but today I would like to focus on RNA or the transcriptome. And why do we use digital transcriptomics, mainly on the nCounter NanoString platform? First of all, because it doesn't require amplification, which is a possible bias, because we're not amplifying, we're actually counting. So this gives us a digital precision. By counting, we also have several logs of dynamic range, and we can go very, very sensitive. We usually go to the femtomolar range, which is quite impressive, I feel. Also, it can be highly multiplex. We can actually capture 800 targets per reaction. We can specify them to the specific target, which can be either immune RNA or a viral RNA, as I will detail later. So we actually do this with very simple fluorescent barcodes. We just have a reporter probe and a target probe, and with these two mixed together, we can actually define our target in a simple and fast and automated way. As you can see here, it's just all very robust, automated, and we get our count. So we just count the barcodes, and this means that we have all these targets, whether they are hosts or virus RNAs. So what did actually this teach us? These are results that have been just previously published, and thank you, Danny, for citing our paper. So this is the first study of this real-world cohort where we had 48 patients and carefully matched controls. So this is a very busy slide. This is called the volcano plot, but I'll just take you through it step by step. So in fact, what you see here on the right side in the red is all the viral and host immune RNAs that are higher in long COVID. On the left side is everything that's higher in the controls. And as you can see, marked in red, first of all, not maybe surprising to a large part of the audience, we found several SARS-CoV-2 RNAs. So the virus is actually present in the blood. However, I should add that all of these patients and controls were at the median of more than 2 years after their acute COVID infection. So this is quite surprising to still find remnant viral RNA after roughly 2 years. Besides these viral RNAs, one of them that was caught our attention was the antisense RNA, which is a marker of ongoing viral replication. And maybe a tiny surprise, we didn't find any spike RNA in most of the patients. We also found, also coded in red, the associated host proteins important for viral replication. And last but not least, for the subject of this talk, we also found the protease, the viral protease, Mpro, which is the target of Paxlovid, currently in several clinical trials worldwide. In green here, you can see, again, building on what Danny said, we see immunological memory being formed. So we see memory B cells that are increased, but also we find transcript for IgE, which is strongly linked to allergies and asthma, which of course resonates with sudden onset allergies that many patients are reporting. And then finally, marked in yellow are platelet genes. Of course, several previous publications have shown a link to coagulation in long COVID, but also the group of Bomzell in Paris have shown that platelets and megakaryocytes can be a viral reservoir. This is quite an interesting observation. This is all quite very busy. So let's just look at individual values first of all the viral RNAs. So here we plotted in red all the long COVID patients, 48. And as you can see, there are very strong differences, but most of the patients had very high levels of several of these viral RNAs. Again, the strongest one was the antisense. We do find other ORs. We found the nucleocapsid. We found the protease. We found a bit less of the envelope, but we find very little spike. And as you can see, comparing to all of the controls here, you see there are very tiny blips of viral RNA in a few of these patients. When we saw all of this up and we look at then what we call the total blood viral load, we see that 60% of the patients have actually a total viral load above the cutoff, which we show here, and only 1 in 12, so 8% of the controls had just slightly above cutoff viral load. So that's what the viral RNA is quite surprising, but what now if you look at the whole immune system? So in fact, what we did is translating this very busy slide, all these 800 genes in biological pathways, which are just easier to grasp. When we look at the immunometabolism, for instance, you can see here in green, a very homogeneous immune response in the controls. And I should add, these are very carefully matched controls from the same general practice. They are not only matched for age and sex, but also for the time since acute COVID, vaccine doses and comorbidities, which I believe is a strong point to make. On the other hand, if you look at the patients, either for the immunometabolism or for the lymphocyte activity, you see a very heterogeneous immune response. Some of them look like the healthy controls, but roughly half of them are deficient, at least in their scores of their immunometabolism and lymphocyte activation. So, biomarkers. So we do believe that we have at least a candidate diagnostic biomarker using whole blood. So this is what we typically use in statistics, which is called the ROC curve. And you can see here, this is a significant result for just the antisense viral RNA. We classify roughly 70% of the patients correctly. We can do also by quantifying an immune RNA fin, but if we combine the two together, which is what you see in purple, we can actually identify long COVID patients with over 90% sensitivity and specificity. But as I said, we do need several flavors of biomarkers. And this is, again, giving the patients the word. So we look at their scores for anxiety and depression, so self-related anxiety and depression over the last 2 weeks. And as you can see here again, these are patients with low scores, meaning they have low anxiety and depression. And this is a log scale. So these are the patients who have the highest level of self-reported anxiety and depression. You can see there's a very strong increase in the virus. And again, there is a drop in their metabolic score. So I provocatively would like to call this anti-stigma biomarkers, because it means, in fact, that the anxiety and depression these patients relate can actually have a underlying biological cause, and patients can no longer be told that it's all in their head. There is, in fact, a biological plausible reason linked to their viral replication and or metabolism. So if we take the statistics a bit further, we also did a multivariable logistic regression to define which of these patients actually have low or viral RNA. And surprisingly, although we know that middle-aged women are the larger subset of patients, age and sex do not influence the high versus low RNA we find in the blood. However, comorbidity is a positive predictor. Each comorbidity by itself raises the odds of having high viral load by 61%. On the other hand, we've talked a lot about vaccination. Each vaccine dose lowers the odds of having this high viral RNA by 64%. So now coming to the clinical trial, what can we say about this protease and targeted by Paxlovid? So first of all, this is a larger group of patients. These are unpublished results. We found that, in fact, we do see the protease expressed in large amounts in the blood. And actually, the protease is very strongly correlated not all, but most of the viral RNAs, again, not to spike, quite surprisingly. Then we look at the the receptors and co-receptors, which are usually absent in the blood. We see that the ACE2 and TMPRSS2 receptors are very strongly correlated to the viral load. They are rare on leukocytes, so maybe we cannot exclude that these could even be circulating endothelial epithelial infected cells. So again, giving patients a word, we looked at their clinical status, and this is their self-reported fitness score. And again, higher scores means patients in worse condition. And we found an even more striking difference. As you can see, these are patients with reasonable fitness levels. As I said, the cutoff is roughly around 10. Most of these patients with reasonable fitness had almost no viral RNA detectable, whereas patients with the lowest score, so worst clinical condition, had actually a lot higher levels of this protease RNA. So probably we can target this clinically. So for time's sake, I'm going to skip a bit over the more nitty-gritty of the molecular details, but we can discuss this further. We basically asked the question if Paxlovid can actually target or modify any of these 4 molecular and clinical targets. So this is where we come to this really patient-led design, what we called grassroots clinical trial. So of course, we should mention that this Paxlovid was used off-label. So this is a very short correspondence that you might have seen. There's been a lot of hard work by general practitioner Mark, by patient expert Elena and Tommaso. In the end, our 600-word correspondence was transformed less than 200 words, but I think it actually captures the essence, and we're glad to have this out. So we do believe that clinical trials are the holy grail of medicine, but they're just too complicated, too expensive, and too slow, meaning that only large, well-funded academic centers usually can engage with the problem. So we propose that clinician-patient pairs, especially general practitioners, are underused in clinical trials. So an online platform was actually designed by the patient themselves. So we made a list, they made a list of long COVID symptoms for each patient that they could use from a drop-down box, and I'll show you an example later. It was the development of an online cloud-based platform that could give us real-time information. And the primary outcome was a drop in total symptoms over 15 days of treatment. So results now. As you can see here, this is a heat map, and you can see from far that is actually a color scale. When you see red, the patients are doing worse, green as the patients doing better. And this is their baseline. So the patient actually can choose from a drop-down menu where they have fatigue, post-exertional malaise, insomnia, brain fog, headaches, etc. I think all very recognizable. So this is their baseline, and you can see how they actually change over time. This is a non-responder. As you can see here, this patient actually had no clinical benefit from using Paxlovid. On the other hand, this is what the responder looks like. As you can see here, fatigue, brain fog, etc., are a bit lower, but still are getting totally green over the course of treatment. And at the end of treatment, this patient had more than 80% improvement of the symptoms. So just summarizing this also, this online cloud-based platform gave us real-time info and allowed us also to identify 2 adverse events, which was important. And our primary outcome showed that was an average 32% drop in total symptoms, as you can see here in this graph below. These are the patients listed before treatment and after treatment. As you can see, some of these patients drop very nicely. You can see those with high scores, meaning doing worse. And also you can see there is a very large variation. Some patients reported low scores, 20, but still had improvement. Others showed very high scores, over 80, but still that is a quite drastic improvement. While all of these patients here in the middle actually had almost no relief from Paxlovid. When we put this in a percentage of improvement, you can see that if you measure at day 15 or measured during the whole course of treatment, there was a quite significant drop in patient-reported outcomes. So currently we have over 147 person months of follow-up. That's the advantage, of course, being based in general practice. So we do have a very long follow-up of these patients. So, of course, we talked about biomarkers. Can this Paxlovid treatment now actually revert the molecular LC phenotype that we found? In short, yes, at least partially. So after the Paxlovid treatment, we found lower viral RNAs, nucleocapsid, ORFs, but not, as you can see here, we did not find a difference in the antisense RNA, which might suggest that it still might be replicating virus and could also possibly explain Paxlovid rebounds that have been shown worldwide in several clinical studies. We also found lower SARS-CoV-2 receptors, and we also found that its treatment reverted several of the long COVID RNA changes. As you can see here in green, there was an increase in total leukocytes, probably neutrophils. As you can see here on the red scale, several of these markers, including our top biomarker FIN, were actually significantly decreased following Paxlovid treatment. So can we do more grassroots clinical trials? Please. Yeah, we only need funding. That's all. So, of course, it's important to highlight some of the strengths and limitations of this study. I think it's important to say as we're a real-world cohort, it's general practice-based for both the patients and the matched controls. We have fully detailed electronic health records, which are national in Belgium. So we have a really unique healthcare record for each individual patient. On the other hand, we don't have data on treatment adherence or previous over-the-counter treatment, which is relevant, of course. We didn't have a sample at the acute COVID, but we did have baseline samples before Paxlovid treatment. We used validated clinical scales for both the clinician and patient-reported outcomes. We had a uniform treatment scheme for all patients. That's really important because all of these patients were prescribed the same first-line treatment, which is low-dose vitamin C, paracetam, clopidogrel, so anticoagulants. And those that opted for Paxlovid were used as second line after this first-line treatment. As I said, this is an opt-in. So we didn't select the patients. They choose Paxlovid treatment themselves. And we also don't have a placebo comparison, which would be important, of course. So if I may conclude, what can digital transcriptomics show us in long COVID? First, I do believe that we see viral persistence in the blood, which is, again, evidence for a systemic rather than a respiratory disease. And it's important to stress that it was 2 years after acute COVID in both the patients and controls. So I do believe this is a possible diagnostic biomarker, which of course we need to validate independent cohorts, which we are doing, by the way, but more are, of course, welcome. We're happy to collaborate with people worldwide. We were able to validate this already in a small number of samples from the Pasteur Institute. We found the same viral RNAs in long COVID patients from Paris. So in agreement with Danny before, I do believe there is a disturbed but not totally immunosuppressed system. We have very tiny but important differences between patients and controls. And what striking to us was a lower immunometabolism, which was correlated to the viral load. So we do believe that we have at least a candidate diagnostic biomarker for long COVID, a compound of a viral and immune RNA. And then we do also have a set of predictive biomarkers for both first-line and second-line treatment. And importantly, they're all non-invasive and affordable, which means we can do blood draws before and after several treatments. We can also look at large populations or cohort studies. So, of course, last but not least, definitely need to acknowledge all the people who helped this. And first of all, definitely to the whole MJ team, but especially to Mark Chamul. He is 77 years old, doing 2-hour-long consultations with the energy of a 17-year-old. So thanks, Mark, for your energy. All the patients and controls, and especially the Belgian Patient Association, I would like to highlight just here, especially Elena and Tomaso. Colleagues from the Rega Institute, you can see here first author Soraya Menezes, as she's actually fighting another viral neglected disease, HTLV, and especially our colleagues abroad from Paris, Dominique Lisa, Peter Brodin from Karolinska, and Jean-Laurent Casanova. And all of you for your attention. Thank you.
Thank you very much for your presentation, Dr. van Weyenberg. Let's start with the Q&A with the first question from the community, which is, how as a patient can I support my treatment and diagnosis with my doctor? It's complicated for non-medics, but in rural areas, you can't always go to specialists, or your doctor or GP simply does not have the capacity to go all in for you.
No, I totally agree. This is highly relevant. So that's why we wanted to highlight the importance of patient clinician pairs. You need to have a GP that preferably that actually listens to you, listens to your symptoms. I think that's the most important thing. And that can actually coach you or mentor you in going to, as Danny mentioned, there are lots of tests sold online, which are incredibly expensive. So I think it's definitely a role there for the GPs in coaching their patients in which blood types or which immunological tests or viral tests to get.
Okay, thank you. The next question from the community is, what about high levels of HLA-activated T lymphocytes? I see them in children with long COVID very often.
Yeah, interesting question. So I'm an immunologist, so I do love these expensive 15-color flow cytometry experiments that can actually show what you are doing. But since we're trying to work affordably, we take in fact the whole blood sample. And of course, this doesn't allow us the real fine-tuning on very specific T lymphocytes. So our cohort doesn't have these specific data. So, but overall, we take the whole blood sample, we don't see that signature. We do see a lower lymphocyte activation overall in the blood sample. But again, we cannot pinpoint that to very specific CD4, CD8 cells or other smaller subsets.
Thank you. Let's take another question from the community now. Okay. What is your view on long COVID potentially being multiple diseases or consisting of distinct subgroups, which may require different biomarkers? And if so, how to navigate this to reach a scientific consensus on biomarkers?
I agree, we do need a scientific consensus. So there, as mentioned before, there are lots of biomarkers out there, 40,000 papers, and each of them proposes its own biomarker. In my personal opinion, I think everyone's right. I think coagulation, clotting is important, serotonin is important, inflammation is important, viral persistence will be important, lymphocyte activation is important, and our metabolome is important. So I do think they're all related and most probably correlated. However, what I always say to my students, if shoe size is a statistically useful biomarker, we can use shoe size. So I do need to feel that for now, we need to be very pragmatic and look at the best performing biomarkers, first of all, at the diagnostic level, because that's an urgent need, because patients are still being told it's all in their head. If we can get just an umbrella diagnostic biomarker saying, well, this is actually with 90 or 95% certainty along COVID, then I think this will help progresses as patients, researchers, and at society level. But then, of course, I do agree it's a complex, multi-system, multi-tissue disease. And in a second term, let's say, after we have these diagnostic biomarkers, we do need more specific biomarkers, which can then guide treatment in these very specific subsets, so that we actually come to this personalized medicine approach that we all want. So treat their mast cell activation, treat their clotting, treat the lymphocyte activation, etc. So I think in short, it's a two-step approach that we need.
Okay, thank you. We have time for a couple more questions. The first one here that's coming up is, are the biomarkers that MECFS researchers have been working on being considered for long COVID?
In short, yes. We just started recruiting also ME-CFS patients because they largely overlap. Some of them actually have been diagnosed retrospectively as probably had CFS before. And we also, of course, want to look at the very current herpes activation and reactivation in these patients, ME-CFS as well as long COVID. So we do want to start looking at them. We just started recruiting. No data yet.
Thank you again. Here's another question from the community. So your opinion is the reservoir of the virus stays in our bodies? What I still don't understand, like MECFS, our body is knocked out. And does this mean this reservoir is responsible? And then how do we get rid of it from the body? As a non-professional, you would think we know how to retrieve it or get it out.
I think it's a very relevant question. So as I said, I can only give a data-driven response on long COVID, which is what we've been studying. So we don't have any data on ME-CFS yet. So definitely we've seen several other viruses, HIV, HCV, all the herpes viruses. Most of them cause lifelong infections. We don't know that yet for SARS-CoV-2. We see elimination, we see a drop over time also in most of the long COVID patients. So I think there is good hope that we can get rid of the reservoir. I think antivirals are one way to go, but probably combined immunotherapy, combined anticoagulation, usually combination therapy would help better. And if you look, for instance, at HIV, there is definitely a reservoir, and after decades and billions of dollars, we are getting closer to understanding, but not getting closer to actually getting rid of that reservoir. So again, more investment, more funding, please.
Thank you. I think we have time for one final question now. And that is, what is the cause of fatigue looking at virus functioning? How come MECFS and long COVID have a crossover? Is there a marker or data overlap in both conditions? Is it true the mitochondria does not work well? What is the mechanism then?
Oh yeah, I should be totally honest, I don't know yet. So we just looked at the overall, as I said, the umbrella diagnostic biomarkers, long COVID versus controls. We are now over 100 patients, over 130 controls. We can now start dissecting the different symptoms. And of course, fatigue is a very important one. Again, we just started to recruit MECFS patients, and we're now analyzing the data of like over 100 long COVID patients, look at these very specific symptoms, which again are largely overlapping, and fatigue is definitely the most important of them. And what I can say at least is that our data show that RNA expression of the virus after 2 years in most of these patients is higher if they are more fatigued, have lower fitness levels.
Okay, we actually have about 2 minutes left, so we'll take one more question here. And that is, how accurate does a biomarker have to be? Sensitivity and specificity. And at what sample size before national health systems roll it out so we can have a validating diagnosis?
Oh, wow. Thank you. That's the excellent question to ask, really. So, I just believe we discussed it in our very short correspondence that over 90% sensitivity and specificity would be ideal to work with. And of course, we need validation in larger cohorts. We have now a larger internal cohort where we have validated this. We are working together with, as I said, the colleagues from Paris. We would love to, of course, validate in other international cohorts. So please, this is a call for collaboration. Please send us your samples. We're also willing to share our samples to look at your candidate biomarkers. I think we should definitely collaborate as much as possible on a large scale.
Okay, we have now reached the end of the Q&A session. Thank you again to Dr. van Vayenberg and to Professor Altman for their presentations, and to the community for submitting questions.